So today we have with us Doctors Richard Gunasekera and Emanuel Tundrea to discuss their recent PLOS ONE Lab Protocol article entitled ORFanID, a web-based search engine for the discovery and identification of orphan and taxonomically restricted genes and the associated protocol in protocols.io. Professor Richard Gunasekera has enjoyed a 20-year career in higher education as a professor and as a scientist in the field of biochemical genetics and nanomedicine. He earned his bachelor's in biochemistry at Baylor University, where he researched and published in organic synthesis as an undergraduate. Richard earned a master's degree in bioorganic chemistry from the University of Houston, Clear Lake, a master's in molecular genetics, and a doctorate in biomedical sciences at the Baylor University Medical Center in Dallas. Before joining Biola University in California, USA, Professor Gunasekera founded the Department of Biological Sciences at the University of Houston, Victoria, and acted as the department head and later as director of graduate studies. He has held faculty and research positions at Texas A&M University Health Science Center, the University of Houston Clear Lake, and Rice University in Houston. Richard's research now spans several interdisciplinary fields such as cancer biology, forensic DNA studies, nanomedicine, and biochemical genetics. He has also received awards for excellence in teaching, research, and as a distinguished faculty member from his previous institutions. Dr. Emanuel Tundrea comes from Romania, where he teaches at Emanuel University in Oradea since 2001. He completed his PhD in software engineering in conjunction with the University of Nice in France in 2009, and then his passion for bioinformatics helped him win a Fulbright Visiting Scholar Award in 2021 under supervision of Professor Richard Gunasekera at Biola University in California, USA. Since 2010, Emanuel has been a member of the Cambridge Scholars Network and also a member of the steering committee of the European Leadership Forum. In 2013, Emanuel was invited to teach a modular course at several universities in the United States. He has been the manager of the Informal School of IT in Romania for 2 years and chairs the IT School of Emanuel University. His research interests extend beyond bioinformatics and has published papers in the areas of ethics of IT, artificial intelligence, and big data. Richard and Emanuel, thank you so much for talking to me today. So to kick off the interview, the conversation, can any of you explain briefly what this protocol is about, how to use it, and why it is relevant?
Yes, thanks again for the introduction, Gabriel, and thank you so much for the opportunity to come and talk about our protocol. This protocol that we simply call ORFanID is actually a algorithm and a search engine that enables the user to be able to find what are called orphan genes across genetic databases, as the ones found in the National Institutes of Health. The well-known one is called the NCBI GenBank, and there are other databases too. So it's basically a search engine, and by inputting a DNA or a protein sequence, you can find it or using a gene ID number, you can find these things that are called orphan genes. So that's the basic purpose of ORFanID, the program ORFanID. We can, you can find it on the web, and I think Emanuel will show us the interface in a minute. But you also asked how to use it and why is it relevant. So I think if I don't define what orphan genes or orphan means, there is not much relevance about the protocol. Generally speaking, an orphan gene is a gene that does not have any homologies with other genes. So we all generally believe in biology that genes are having some homologies with related species or related genus, different families and orders and phyla across the taxonomical tree. But these orphan genes are a new discovery, relatively new, where they are not having any relatives or ancestors. They're orphans, meaning they have no homology. So what's unique about that is this particular program can go across different species and different taxonomical levels in the phylogenetic tree of life and find if it's only restricted to a species, the gene, or it might be even restricted to a taxonomical level, which we call a TRG, or a taxonomical restricted gene. So it's a very easy to use web-based intuitive program that any biologist or a geneticist can go in and find. So one last thing on the area of relevance, I think it's important to see that this is going to help scientists, biologists, medical people to learn more about our gaps of knowledge in genetics and genomics. And so I think this is very significant because it might even help users and others, medical practitioners, particularly researchers, to be able to learn about diseases, not necessarily genetic diseases alone, but even others that may have a genetic origin, such as Alzheimer's, diabetes, even such disorders, by learning about this particular area where there is so many gaps, and even understanding the formation of brand new genes, evolution, because we call these genes de novo or de novo formation, meaning they are formed new.
I see. And actually in the paper, you do a pretty good job at describing what was the state of the art when you embarked in this project. And you describe, other existing platforms or algorithms that could help identify these orphans' IDs. But can you briefly again summarize, to understand the relevance, what was the state of the art and why you decided to develop this particular algorithm and web-based tool?
Why don't I let Dr. Emanuel Tundrea give a stab at that question first?
Absolutely.
Hey, again, I want also to thank you for this invitation. It's a very big privilege for me to be part of this wonderful team. I'm, in a metaphorical way, I'm an orphan among these authors, because as you previously underlined, I come from an IT background, so I'm not a biologist. But this team of professors and researchers really fascinated me with this project because, as Professor Richard Gunasekera underlined, it opens an amazing world of understanding of and research to see why we have this de novo, as they are called, or these orphans. And to my surprise, to see that the percentage of the orphans in any species is quite high. And even if the databases are growing and more and more species are sequenced, it seems that there is everything reaches a plateau. The number of orphans stays. Now, coming back to your question, it was fascinating for me to see that there is so much computational work to be done, and there was a big need for an efficient algorithm to identify these orphan genes, since the scope and the volume of data in genetics is growing so much. There was a wonderful opportunity for me to play with the data and to see, provide statistics and ways of identifying or optimizing things. But I also relied on the biologists to interpret and to see. Now, there were some attempts before, and we mentioned in our paper Orphan Finder. This tool was probably the most close in the scope of our initiative. The problem was with Orphan Finder is that it stopped its support in 2016. No longer, the tool no longer works, and its scope was limited, only worked for proteins and only for plants and fungi, not for many other species in the tree of life, and not working for DNA, for nucleotides. ORFanID has a wider scope. It provides the opportunity to search orphans in a given species, both for DNA and for proteins.
That's a very good point. Yeah, the scope was very small. It wasn't very intuitive. You have to compile. It's more like a tool for computer scientists. We wanted to prepare one biologist, and non-computer scientists can also use. So that was the big difference, and our scope is much greater.
Yeah, and the beauty of ORFanID is that it has a web interface. So no more black screen, installing some driver and writing a command line. It's user-friendly.
It's user-friendly, exactly. And I'm not a computational biologist myself, so I don't know how relevant this question might be. But while you were developing the algorithm or the protocol or the tool, was there anything that you tried as an optimization but didn't work and potentially didn't make it to the final version? Again, I don't know how to write code, so I don't know whether the question is relevant.
It is, right, Emanuel. We did, during our development, we tested different methodologies of approaching, because we are using also other algorithms embedded in NCBI called BLAST. So it's a BLAST-centered technique. So our earlier algorithms, were not as versatile. And in fact, as a result of peer review, which is a wonderful thing, we also incorporated another algorithm called PSI BLAST, which, so that helped to optimize our protocol and refine it and bring about more information. Emanuel, you're welcome to say more if you like.
Yeah, actually, I during my Fulbright, I stayed there for 6 months, the first 2 months. So quite a heavy, I got a big portion of my time. I just invested in testing other software, other tools. So there are Diamond, MMSeq2, Orphan Finder. I was trying to use it. So as in any research, the first part of doing the literature review takes so much, just selecting the most interesting articles. This was also our very interesting and it took quite a long time to identify the third-party tools that might help, not having us inventing the wheel. And also BLAST, we chose finally BLAST because BLAST is an authority and people are recognizing it as a good tool.
And for the new user, what are the most critical steps when using the protocol successfully for a successful use?
Could we share the user interface? It might be,
Absolutely.
yeah.
Oh,
So
sure.
it only the video only shows who's speaking.
Oh,
So if you're gonna show the interface, you'll have to explain. So let me allow screen sharing options. Now you should be able to share, whoever wants to
Yes.
share.
Yes, it works. Thank you. So if my screen is visible, the interface is quite intuitive. The steps are presented in our protocol, and there is on the home page, there's a video explaining how this can be used. Now, since you asked what were some problems for a new user, I tell you that there were some very common and even myself, I did quite many times this problem. The interface accepts either a FASTA file or of a sequence or a series of sequences or accession numbers from two biggest databases, NCBI and UniProt accessions. Now, I would ask our users to be very careful. If you insert here an accession number that has to do with a protein, you have to have the selection on protein.
the radio button, yeah.
Yes, because if you check a gene, a DNA, and you insert a FASTA file of a protein, then actually you check a protein against the whole DNA, and the result will not be good, and vice versa. And there's one more thing. In order to do correctly the classifying, maybe one sequence is not an orphan, maybe is a TRG. And this is another beauty of this tool, is that it works also for taxonomic restricted genes. It tells us if one sequence has homology at the level of of genus or a family or an order or up to the super kingdom. Now, if we enter an accession number that belongs, let's say, to Homo sapiens, and then we select the organism as E. coli, the classifier will have a problem. Now, we try to find this error spot on and give a feedback to the user, but sometimes it may happen that this comes through, and then it says that it's an orphan just because this sequence or this protein it's find in which is in a Homo sapiens cannot find in E. coli or in another organisms. This would be maybe the two biggest.
Yeah, so you have to choose the correct organism when you put in your sequence of the DNA, of the protein, or the accession number of the gene. So the gene ID also is another word that's used. And those are found from the NCBI. And, a person has to be familiar with that if that's what they're doing. So, yeah, it's straightforward. And we also have a video that explains that. Plus, protocols.io guides you how the whole protocol should be followed.
It's a beautiful interface. I'm not a geneticist or a genome biology myself either. So, but it seems, it seems powerful, and the interface is very user friendly. So a lot of work is behind the development of this tool. Why did you decide invest even more time and have this as a standalone methodological paper formally peer-reviewed in PLOS ONE? What was the motivation?
So peer review is kind of the gold standard of good scientific research. And this particular tool is for researchers. And I would be remiss if I don't mention the fact that we had a team of scientists working on this, and some practitioners and some scholars and academics. So for us who are academics particularly, this is important, and for funding purposes and other things. So we wanted to publish not just the protocol and its operation, but also the algorithm, so people can understand and learn from that and build their own algorithms. So we give access to all of that information, and that's why we wrote it up as a methodological article. And in that, there are 5 researchers. Dr. Paul Nelson is one of them, who's a great science philosopher, and he's involved in the next step of this, which we call orphan base. We are building up a database. And then we had programmers from the UK, Suresh, and then in Houston, Texas, the bioinformatics group, Vinod is one of them. These are people who have doctorates too. So it's very handy to have Dr. Kumar also from the Baylor College of Medicine. So just wanted to mention the other authors also involved in this. But we wanted, for their purposes and the furtherance of our work, and mainly more than our work, but for other researchers to gain knowledge and information, we wanted to publish it and go through peer review.
And you previously mentioned that in response to the peer reviewers, you decided to include SciBlast. Is that correct?
Yes.
So
yes.
Was there any other feedback from the reviewers that helped shaping the final version of the protocol and the manuscript?
Emanuel, can you think of any others? There was quite a bit of good feedback.
Mm-hmm. Yeah, so you mentioned the scientific legitimacy of having a peer review article out there. We also hope, and again, thank you for this opportunity to present this live on a video. We hope to gather a community of biologists and computational bioinformaticians, and so we get this feedback and continue to refine. There was yeah, I can't remember specific. There was, to be honest, there was one orphan. There was one sequence that our ORFanID proved, classified as an orphan, and actually it wasn't. And we found the mistake because we didn't update to the latest database of protein and DNA. And since then, we kind of made a rule that as soon as as soon the database is growing and there's a new release on NCBI or anywhere else, we update our own server. Yeah.
I also remember another peer reviewer mentioned that this could even be, the program could be used to identify or use it for forensic purposes. So it's for investigations of illegally exported animals or species by running their genes or their DNA across this program. So we didn't incorporate all of that information, but these are all new ideas that we didn't necessarily think of it ourselves to use it as a forensic investigation for DNA purposes of illegally exported animals and other species, even plants.
I see. And well, for anyone who's curious, actually all the information that it's in the manuscript itself, because you decided to make the peer review history public. And then that's my following question. why? What was the motivation to have all this conversation between you and the expert who read the manuscript before publication available?
Yeah, I think transparency is very important. And we really like PLOS ONE, the journal, for their level of transparency and their measurement and assessment of good scientific work based on the science mainly and the procedure of the scientific method. So in the same manner, we wanted to be transparent and let any reader learn from the rigorous peer review we went through. And in addition, maybe students and learners who hear about this so-called peer review, what do you do, how to publish a paper, will see for themselves the kind of questions peer reviewers ask and how we have to respond and how it's done in a scholarly and a gentle manner. So I think those are the main reasons we wanted to make the peer review transparent or available.
And my last question is, you both come from, very different angles to tackle this problem, have different career paths, located in different institutions in different countries. And I know this is a relatively young paper, but could you speculate or think ahead? What will be the significance of having developed this tool and published this protocol and paper for each of your careers?
So if you like, I'll start and let Emanuel follow. So actually, this is the beginning of a tool that we wanted to find to identify orphan genes so we can build this huge database of orphan genes. We call it ORFanBase with the letters ORFANBASE.com, orphanbase.com, where we've already started building a database of orphans from different species, again, according to the phylogenetic tree. So it's a very interesting thing to go and see how many orphans are found in the human or in the E. coli or plant and things like that. So it is the beginning, it's the tip of an iceberg, if I may say. So, and the different scholars who are involved, who are interested in orphan genes can now go to orphanbase.com and look at it. And I think it's also helped career-wise in different ways, because probably we will publish some of that orphan-base related work in a journal like PLOS ONE. And I had the privilege of having Emanuel Tundrea come from Romania as a Fulbright scholar. So that helped our university. I think would help Emanuel's career too, because he's a Fulbright scholar. And new Fulbright scholars might also be interested in doing this kind of work. So their careers will be enhanced. Our universities were really both profited as a result of this by having the exchange of scholars. So I think it is very helpful for our careers too, because we can go on to build other information bases, such as the orphan base. And for orphan base, we're actually even using AI. That's part of our new approach, to use artificial intelligence in identifying orphan genes. And so this is both DNA and protein.
Well, I cannot, I can only echo Professor Richard. I was really blessed, and it means a lot for me. As a young researcher coming from Eastern Europe, and having this opportunity to be in this community of researchers at Biola and Discovery Institute and others who came alongside and helped me. I was helped also in my English proofreading, as you can see I'm not a native speaker in English, and it helped me a lot to proofread my writing and to learn methodologies, how to publish. It has been, I felt like a disciple in this team, and career-wise, Fulbright has this goal of building partnerships between countries. And for me, this is one of the privileges to continue even after the program and after the funding and everything, we still continue working together. And as Professor Richard Gunasekera underlined, we have plans for refining this tool and also building the orphan base, the database. And so there's hope for future.
Thank you so much, both of you. They have been like a really illustrating conversation. And just before we finalize, is there anything that you would like to mention regarding the protocol, the process that I haven't touched, that we haven't covered?
I think we covered most everything, but I do want to again acknowledge all the different computer scientists, bioinformaticians, and Biola University, Emanuel University, and then also Fulbright fellowships and Discovery Institute for all of their support. We were really well supported and very thankful for all of that.